We are going to, again, start talking about neurons today. We'll get different types of neurons, different parts, breaking down the parts of the neuron. You're going to keep them. Yep, we're going to change up the mode. So your assessment, your quiz is going to be your points pertaining to your completing that. Yep. We're going to try that out. If it doesn't work, I'll go back and revert to my old ways. But we'll try the assessment to start off with. Okay? Got our objectives. Now, our objectives posted up there are for today and tomorrow. 2 out of 3 of them are really tomorrow. Okay? So the first one, describe how message is transmitted through the nervous system. We're going to get to that today. Going to break down the neuron. That helps us to figure that one out. And then the second and third one, neurotransmitters, summarize what they do to contribute to the nervous system. Kind of already hit on that just a little bit with acetylcholine. And then provide examples of the functions of major neurotransmitters. Again, we're going to break that down a little bit more tomorrow. Okay? Just want to summarize this idea. This is what we've been trying to get at with our bell work assignment today. It's really, before we can understand human behavior and mental process, before we can understand psychology, we must understand the biological connection. Okay? Must understand the biological connection. I do want to reiterate that I'm going to put a lot of stuff up there on the board. Sometimes when I put this together, I put too many words up there. But that's just to initiate my thoughts. And what I encourage you to do is to prioritize the thoughts that I verbalize rather than the notes that are written up there on the board. Okay? And I do go through them fairly quickly. Okay? The neuron. There are 3 types of neuron. First off, the neuron is defined as the brain's most basic, or the body's most basic thinking cell, because really when you think about it, the neuron not only encompasses the brain, but also your nervous systems as well. And when we get to the spinal cord, I'll mention this I guess a little bit later, but the spinal cord is actually an extension of the brain. And so if we think of it as an extension of the brain, then it can actually make decisions on its own. Sometimes we don't think of the spinal cord like that. But there's a reason for that. And again, we'll get to that a little bit later. Right? So 3 types of neurons. We need to be able to differentiate them. There's afferent neurons, efferent neurons, and interneurons. First off, the interneurons are kind of like the in-between neurons, the connecting neurons. Okay? And essentially, what are they trying to connect to? When it's part of the nervous system, like your peripheral nervous system, it's trying to connect it to your spinal cord. So then it can go up to your brain. Okay? So think of interneurons as connecting neurons. They're the intermediaries. Now, afferent and efferent. This is where students get mixed up. Right? They confuse these. Okay? So I'm going to give you a suggestion on how to differentiate them. Afferent neurons are your sensory neurons. So we got 5 senses. Go ahead and tell your partner real quick. Remind them of the 5 senses that we have. All right, so you look at this guy up here, right? What sense is he focusing on? Smell, right? He smells something, okay? So your nose, your mouth and tongue, your skin, your eyes, your ears, all of those are really receptor sites, okay? And since they are receptor sites, the stimuli that you're exposed to, the neurons at these receptor sites are going to be afferent neurons. Now, those afferent neurons aren't going to connect directly to your brain necessarily. They're going to be connected to interneurons that eventually are going to work their way all the way up to your brain. Sound good? So it allows us to interact with the stimuli in our environment, are our afferent neurons. Okay? Now, our efferent neurons are our motor neurons. So when I think of motor neurons, I think of movement, muscles. A lot of M words, okay? Muscle, movement, motor. So whatever gets me up and going, that's my motor neuron. So you've got pencil to paper, moving the pencil across the paper, that's your efferent neurons is going to enable you to do that. Okay? So one suggestion that students came up with a long time ago was it takes effort to move, right? So you think about effort and efferent neurons. It's an easy way to make another connection there and differentiate them from afferent neurons. So I'll give you a scenario. Say that you're doing some construction and you're doing some demolition on an old shed, right? So you're taking these boards and off the structure, and some of them are laying down on the ground, and you're walking around, and all of a sudden you realize that you have just stepped on top of a nail, right? And so it has inserted your shoe and penetrated your skin, right? So which of those neurons allows you to feel that nail in the bottom of your foot? That's your afferent neuron. Then what type of neurons allow you to remove your foot from that nail? That's your afferent neuron. Okay. Now again, we don't want to forget about the interneurons. Those are the intermediaries. Those are completely necessary, all right, in order for everything to work out. So this is kind of what looks like, neurons. This is neurons inside of our brain, okay? Or it could be actually neurons inside of our nervous system as well. But you can see the lights moving through there. That's kind of symbolizing the neurotransmitters and this energy passing from one neuron to the next and communicating, these neurons communicating with one another, okay? And we'll break down their structure here in a little bit. Actually, we can do that with our 3 parts, okay? So there's 3 main parts. There's more than the 3, but we're going to start with the 3 main parts of a neuron. There's the cell body, and this contains the nucleus, and so that's what is going to determine really what when the neuron fires, what type of neurotransmitters the neuron's going to produce, whether it's resting or not, okay? All that takes place inside the soma, okay? There are, you may write this out of the soma, there are receptor sites on the soma. It's a commonly mistaken mistake that students make, okay? There are receptor sites. Now, what do I mean by receptor sites? Neurotransmitters are going to be passed from one neuron to the next. In order for them to be passed, they have to have these sites for them to be received, okay? The majority of those receptor sites are actually going to be on the dendrites, part number 2. But there are some receptor sites on the soma as well. Sound good? So the dendrite, in the Greek language, it means tree. So I think of tree or branch-like extension. Okay, so if we go over here on the wall, there's another picture of a neuron. The round structure up here, right, is the soma, the cell body, and that large dot, yellow dot there in the middle, that would be the nucleus. Now you see all these branches off of it? Right, that's the dendrites. Okay, that's the branch-like, tree-like extension off of the soma. Okay, now this long snake-like, tail-like structure, that's your axon, and attached to the axon are these what's called axon terminal buds. Okay, now when you look at the axon terminal buds, sometimes they look fairly similar to the dendrites, don't they? Okay. Now what you have to do to make sure that you can differentiate them is you think about what are they attached to. Okay? If they're attached to the soma, then that makes it the dendrite. If it's attached to the axon, then it's axon terminal buds. Okay? Now, the flow, the direction of energy, the direction of neurotransmitters is kind of important as well. Neurotransmitters are going to be received by the dendrites or the receptor sites on the soma, and they're going to transition this direction. Okay, it's going to go down, right, down the axon into the axon terminal buds, and then they're going to be expelled out into a fluid-filled gap, which is called a synapse, to the next neuron. Everybody understand that? So you may want to, if you're drawing this on your sheet of paper, you may want to draw an arrow, like a direction of which that really helps to give you a better idea of, the dendrites versus the axon terminal buds. Sound good? Okay. So you can see how it went down? I think there. Everybody see that? That's the imagery I want you to think about. It's going, this would be like the axon. These are the dendrites up there. There's the soma. It's going down the axon, and then it's going towards the next neuron there. Sound good? There are 2 more parts of the neuron that I want you to be familiar with. You can see the dendrites here, the axon terminal buds, the soma right here with the nucleus inside of it. The 2 that I want you to write down and add to our list of the 3 is this, the myelin sheath, and then the nodes of Ranvier. The myelin sheath and the nodes of Ranvier. Okay? The myelin sheath is like a white, fatty substance that surrounds the axon. and then your nodes of Ranvier are like these gaps that are between the myelin sheath. Okay? Now, your axons, or your neurons as a whole, your axons of your neurons, when you're born, a lot of them don't have myelin sheath on it. They say not myelinated, right? And at different stages in your life, different parts of your brain become myelinated, or the axons become myelinated. I'll give you a specific example. Right now, with the age of your brain, there's a part in your hindbrain called the reticular formation. The reticular formation is called, its function, one of its main functions is focus and attention. We call it the focus center of the brain. Okay? Now, at this point in time in your life, it's not fully myelinated. Okay? So that means that it's not functioning at its highest level. So if it's not functioning at its highest level right now, what does that mean for you and your attention and your ability to focus for long periods of time? Yeah, it's inhibited. Okay? You guys ever been to Miss Axelson's class? She does a great job. What is of teaching? What does she do with her classes? Yes, yes. I think she even sets a timer sometimes, doesn't she? Right? It's like 7 minutes, 7 minutes, 7 minutes. She chunks it, right? And the research behind that is focused on this idea that your reticular formation is not fully myelinated. That she understands that after 7 minutes, I'm going to lose you. Right? We better move on to something else because we're not getting anything done either way. Sound good? Right? So the myelin sheath, what does it actually do? It actually speeds up the neuronal transmission. So these neurotransmitters that are being passed through the neuron, because of the myelin sheath, it's going to pass through much more effectively and efficiently, much more quickly than it would have if you did not have any myelin sheath there. Okay? So if the teacher ever catches you drifting off sometime, and not being able to focus, they catch you off guard, maybe try to embarrass you or something like that, well, you just reciprocate to them, say, hey, my reticular formation is not fully myelinated yet. How can you expect me to maintain my focus for the extended amount of time that you have in class? And see what they say then, right? I'd like to have a student do that sometime, okay? All right. Another important cell that we have in our brain is the glial cell. I usually attach 2 V words with the glial cell, okay? 2 V words to help us remember its functions. First one, it says brain and spinal cord adhesive. So it holds things together. Can you think of a V word that holds things together? Velcro. Velcro is a good one, right? I know of a little boy that can't tie his shoes. Velcro is a lifesaver, okay? It is small. Glial cells are small, but they make up half the human brain. The other function that I want you to think of is they act like garbage disposals. What other V word do we know of that kind of cleans things up? Vacuums. So vacuums and Velcro is what I want you to remember and think of when we get to glial cells. Sound good? So this is kind of introducing the idea that your brain cells are going to die. Right? You've been reading the article. Some of you already started doing that, right? And you see that the brain weight over the course of your life, what happens to it? It starts to decrease fairly substantially. After what age? Did anybody pick up on what age? It's about age 30. So I'm on the downhill side. Right? So there's a pessimistic approach to that, but the reality is that the brain is still, look at B.F. Skinner and what he was able to do, right? It still has a lot of potential even after you start to lose brain weight. So what is it that helps to clean things up with all these dead brain cells? It's your glial cells. All right, this is a good image to help us to understand the role of glial cells and how important they are and how popular they are. All the green, that's glial cells, right? The red, what would the red be? What part of the neuron would the red be? It's the soma, right? It's the only part of the neuron that's really popping up in this particular image, okay? It's the nucleus, it's the dark area inside the red, okay? All right. Magic at the synapse. This is where it gets to be a little bit more complex. It's easy. You got 3 different types of neurons. You got 3 parts of the neurons, or main parts of the neurons. Now we start talking about the synapse. And this is where I start maybe losing some people. The synapse, you may want to start off with this. It's a fluid-filled gap between neurons. Fluid-filled gap between neurons. Sometimes the synapse is referred to as the synaptic cleft, like C-L-E-F-T, synaptic cleft. I think of it like a cliff, like you fall off, right, the edge, and you're out there as a neurotransmitter, just floating in this small space that's between it and the next neuron, okay? So a long time ago, scientists, they knew that they had these cells, the neurons, and they knew that there was energy that was passing between them. And so the analogy metaphor that they had was that it was electrical. It was all electrical. They thought of it like a copper wire, like energy passes through a copper wire, like electricity passes through a copper wire. But when they started to look a little bit closer, they realized that neurons, they're not physically connected to one another. They're not actually touching each other. So that whole metaphor, that idea, it doesn't work. So they had to come up with a different type of explanation, and they started to realize that there's this concept of neurotransmitters. So when they say magic at the synapse, what they mean, it's a combination of both electrical impulses, and a chemical component or a chemical reaction there, right? The chemical aspect is the neurotransmitter. The electrical aspect is the neuron firing or not. Sound good? So that's why they call it the magic at the synapse. It's both electrical and chemical at the same time. Okay? So down here at the bottom, it talks about permeability in neurons firing. So we're going to go away from this for just a second, and we've got a drawing for you to consider. Okay? The concept that I want you to think of whenever a neuron fires is ions. You got to understand ions before you can understand how a neuron fires. Okay? So we got these chemical substances of neurotransmitters that are going from one neuron to the next. But even beyond that, there's even smaller particles that have these electrical charges to them, whether it's positive or negative, that are floating either inside or outside of the neuron. Sound good? So they're depicted by plus and minus signs here. Okay? So here's your neuron. Here's our dendrites, soma, nucleus, axon, myelin sheath, right? And then here's our synapse. Okay? There's synapse here, and everything around it has ions in it. Okay? Now at rest, I have to look this up because I could be wrong, but I think it has a negative state at rest. So what we're saying here is that inside of the neuron, it's got more negative ions than positive ions. Sound good? Now, when the neuron receives, you got this neuron coming into the dendrite here, and it's coming into the soma, right? And it meets up with the nucleus. And it's classified as an excitatory neurotransmitter. We'll get to those a little bit later. An excitatory neurotransmitter says, hey, we as a neuron, we got to fire, right? Tells the nucleus that we got to fire. So how does the nucleus fire? Well, it's going to match up the number of positive ions or the number of negative ions with the number of positive ions. So there's this concept of permeability. So the cell membrane, it's going to open up. All of a sudden, here come the positive ions. And then once it matches up, if it has the same number of positive ions as it does negative ions, then the neuron fires. Right? And when it fires, it takes the neurotransmitter, goes out of the soma, or all of the many neurotransmitters, goes down the axon, goes faster if you got the myelin sheath, and then to the axon terminal buds, and then spits it out into the synapse there. Sound good? So this is why, and it's a little bit complex, this is why they call it the magic at the synapse. So it's firing, that's the electrical component, right? And then the chemical component is the neurotransmitter that's passing through the neuron out into the synapse. Sound good there? They're going to travel fast though, okay? So you may think that this is kind of a complex process, but it happens fast. says, so what causes the intensity of our emotion? Well, it depends upon the number of neurons that are firing and the rate at which they fire. Right? If you get a lot more neurons firing, then maybe your laugh gets even more accentuated, louder. Right? If your neurons are firing really fast, then maybe the sadness that you're experiencing is going to be more intense. Okay? These impulses that are going through our body with the electrical impulse, the firing of the neuron and the neurotransmitters, this is a misprint here. They travel at a rate of 224 miles per hour, which is aided by the myelin sheath. Okay? So myelin sheath, it speeds up this neuronal transmission. All right? Without the myelin sheath, things are going to break down a little bit. They're going to be less efficient, less effective. Okay? So an analogy that I have with the myelin sheath and the nodes of Rentier. You ever been to the airport before? You seen those walkways? Right? First time I went to the airport, I was kind of your age. I didn't go get on a plane for a long, long time. And I got there and man, these things are cool. All I have to do is stand on them and go all the way across the airport, right? And I looked around, had a huge line standing behind me. Right? I was like, oh, you're supposed to walk on these things. And then you move across the airport exponentially faster, right? That's what the myelin sheath is for, okay? We're out of time today. We'll try to pick up here and talk about neurotransmitters a little bit tomorrow. Maybe clarify some of these ideas of the magic at the synapse. Appreciate your attention today, guys. You guys have a good rest of the day.